righty, I think we'll go ahead and get started on our next roundtable workshop, FT and TD-NMR, complementary for metabolomics. And at this point, I'm going to turn it over to David Rice.
Okay, welcome everyone to whatever this is for our next meeting. I think John knows the number. So without further ado, I'll hand things over to John for a welcome.
Well, thank you, Dave, and good morning, everyone. Good afternoon, good evening, wherever you may be. And Dave, that magic number, I believe, is 52 since we started in 2016 between live meetings and, pardon me, the Zoom online workshops, number 52. So a bit of a milestone, and very happy to have everyone with us today. Very good subject material as always, and as probably most of you know by now, the IVAN meetings, both in person and Zoom meetings, are sponsored by MR Resources and Q1 Instruments. MR Resources, of course, been around for nearly just about 40 years at this point, and focusing on pretty much everything one needs for NMR systems, services, and parts. And as always, they've got a huge focus on service and repair of your NMR spectrometer. They've recently increased staff significantly, engineering staff, applications folks, and support staff to fill in their service contract program quite a bit, as we hear quite more than we should. And I think a lot of people here that or experience regularly that getting service on your NMR spectrometer, even when you're under service contract with either the manufacturers or other providers, can be difficult at best. And MR Resources really is trying its best to put a change to that. Join their service contract program, and you'll get absolutely immediate response to customer problems. They've got the very best engineers and applications folks located throughout the U.S. You'll get priority service and support and scheduling. Obviously minimizes critical downtime. They offer, where needed, testing and certification for GLP, GMP, and other required compliance. And last but not least, very, very significantly less expensive than other providers. Give them a try, get in touch, and I think everyone will be very glad they did. Anyways, down to business. First, I'd like to announce the upcoming IVAN/ENC meeting. That's the day before ENC meeting, Saturday, April 15th, live from Asilomar. We haven't been out there for 4 years, hoping many, or if not most of you, can join us out there. It will be a hybrid meeting, online as well. So those that unable to attend in person, you'll still be able to join in the festivities. Very important component of the day-long IVAN meeting is the annual IVAN Founders Award. And that's going to be, it continues to be a very, very good, very important component of the meeting. The call for nominations, I know, was just released, has been emailed out quite a bit, posted around, and can also be found on the IVAN website. All that being said, I'll turn it over to you, Krish, and maybe you could give a little bit more information on IVAN ENC and also upcoming meetings.
Thank you, John. Welcome, everyone. Just wanted to talk about what is coming down the pike and how you can help us as well. We are probably taking a month off in February, unless somebody is willing to do a short workshop on short order. Keep us posted. If you can reach us by the end of this week, we can probably organize one in February as well. The next workshop is in March, 30th of March, by Roberto Gill. It will be again on residual dipolar coupling and its application. A good panel is being put together as we speak, and it's on March 30th. The following that in April, as John mentioned, we have ENC user conference. It is a hybrid meeting, so as a participant, you can join, you can be there in the room or through the Zoom. So we call it Room on Zoom conference. As a speaker also, even if you are not coming, but you would like to make a presentation at the user conference via Zoom or in person, either way, and you have an interesting topic to talk about, reach out to us. You can reach to me or Dan Iverson or any of the board members, or info@ivanNMR.com, and we will try to accommodate your talk in our user conference as well. It's on April 15th. It's an all-day conference live from Asilomar. Then following the ENC user conference, we have quite a few interesting topics coming down the pike. Perhaps the water suppression method is one that is being discussed. So if you have other topics that you would like us to consider post ENC, do let us know, and we will come up with a plan to provide a workshop on that as well. With that, I think I will pass it back to Dave for today's meeting, or actually John. Yes, John.
Krish, thank you very much. And everyone, please do continue to join us for all of our upcoming meetings if you possibly can. Just one more brief word from our other sponsor, Q1 Instruments. Don, if you would, we'll then get started after that.
Thanks, John. Q1 is dedicated to developing NMR hardware and software for engineers and broad range of engineers and scientists. We invite you to get to know Q1, and the Quantum One Plus is the cornerstone of our platform and has gigahertz intermediate frequency, which provides artifact-free performance and is available as an upgrade to existing spectrometers from 300 to 600 megahertz. We offer flexible configurations with an excellent performance at an unbeatable price. The probe is a hallmark or the key part of an NMR spectrometer, and we offer the smart tune and match probes with a robust hybrid tuning mechanism, faster tuning, unmatched reliability and throughput, and our patented design with free from drift and hysteresis, excellent signal and noise, and superior solvent suppression, made by the Q1 team in Zurich. The probe configurations can be manipulated to your heart's content, and also multi-platform integration for Brooker invariant systems. Want to know more? Give us a call or email us, or go to our website at q1americas.com.
Thank you very much, Don. Very capable product line. Please do have a look. We'll get right down to business. Dave, if you would like to start things off. And Dave, there you go.
Yep, right. Okay. So I would like then to hand things over to Istvan, and why don't we get started?
Thank you very much. It is a great honor to be part of the Ivan workshop series, and thanks for Krish and John and everybody else on the team to make it possible. Welcome everybody, and special greetings to my Hungarian friends and colleagues. I am Istvan Pelczer. I work at Chemistry, Princeton University as a lecturer and director of the NMR facility. Let me introduce my panelists in the order of appearance. Dave Cistola will be first, who is Professor, Center of Emphasis in Diabetes and Metabolism at, that's a long list, at Texas Tech University. And then AaryaniSajja, if I didn't botch the name, too bad, Thank you, who is an assistant professor at Department of Biomedical Engineering at University of Memphis. Just in a short summary, let me tell you how this panel came together. I got this idea of the FT-NMR, TD-NMR combination because of our own work. But when I got the opportunity to organize a panel, Dave Cistola came to my mind at the first place, who, first of all, TD-NMR is on the market for decades, but for industrial and agricultural purposes at the first place. And David was the pioneer who got the idea and published beautiful papers with Robinette in 2017, I believe, when they published the results using TD-NMR for biofluids in regard of diabetes, and then he will talk more about it. When I was looking around for time domain, I had to realize something I didn't realize before, that there is another angle of time domain NMR, and that is using craft. Because craft is a time domain manipulation or analysis of spectra, or more exactly time domain data, directly providing information about spectral features. And it is only just for convenience and being old-fashioned to present the results in a spectral form as well. And I ran into a fantastic paper from Aryani. She will tell you more about it. There are more to say, but I will weave it into my own presentation at the very end of the line. So that's for introduction, and then Dave is first.
Good morning, everyone. So I'm happy to be here, and I'm very much appreciative to Istvan for the opportunity to participate in this. And I'm going to talk today about relaxometry methods and devices for cardiometabolic health screening. So some of the words were carefully chosen here. Relaxometry in this context means monitoring relaxation times in the time domain. There's no Fourier transform or no attempt to work in the frequency domain in this approach. And then the other key word I want to point out for now is screening. A lot of people mix up the concept of screening and diagnosis, and I'm going to come back to that again later. So I'm trained as a physician and a biophysical chemist, and my PhD is in magnetic resonance and related methods. And for the first part of my career, I did a lot of work with high field spectroscopy on proteins and lipid protein complexes. But about a decade ago, we switched our focus to ask the question, what is the simplest form of NMR that might have some clinical utility? And that's how we got started on all of this. And so I'm going to talk to you about that today. Okay. Everyone knows, of course, that the diabetes is a worldwide pandemic, and it's a growing pandemic. Unfortunately, it's not getting better at all. I showed this slide to second-year medical students, and the last version of it from a couple of years ago showed that number is 460 million. It's now up to 537 million adults worldwide that are living with type 2 diabetes, and almost all of this is in adults and the type 2 form as opposed to the type 1 and a few other minor forms. So it's bad enough that we're dealing with over 500 million people, but in reality, this is just the tip of the iceberg. So I'm trying to envision having 500 million people crammed onto that iceberg here, but unfortunately, there's an even larger group of people that are lurking below the surface that have pre-diabetes. And then another condition that comes before that called early metabolic imbalance, which most people have not even heard of, including most physicians. But this is where it all begins. And in a similar way, the same thing happens with cardiovascular disease. That's the tip of the iceberg. And there's a lot of people that have a precondition called metabolic syndrome. Metabolic syndrome and pre-diabetes are, you could say, cousins, maybe even siblings. They're overlapping definitions of the same phenomenon. And then underneath that, of course, is early metabolic imbalance. So the same root cause is leading to both of these, cardiovascular disease being the #1 cause of death in the US and in the world. So both of these are massive problems to address. So EMI includes a combined impact of compensated insulin resistance, adiposity, subclinical inflammation, oxidative stress, and dyslipidemia. That's a mouthful of terms if you're not familiar with medical terminology, but it's not one thing. It's a combination of things that occur together And that's a key aspect to what we're doing with screening. So the key to preventing diabetes is to protect the beta cells, to protect the damage to beta cells in the pancreas. Those are the cells in the islets of the pancreas that secrete insulin into the blood. And so the path to type 2 diabetes is actually quite slow. It occurs over years to decades, and it goes from healthy metabolism into early metabolic imbalance, and then pre-diabetes and or metabolic syndrome, and then finally overt type 2 diabetes. So that's the tip of the iceberg part. In the early metabolic stage, I call this the yellow zone. So we've got the green zone, the yellow zone, the orange zone, and the red zone. So in the yellow zone, the pancreatic beta cells remain intact. The body's trying to fight against insulin resistance. So the beta cells are working really hard to compensate for the insulin resistance in target tissues like muscle, liver, and adipose tissue. But the good news is that you can reverse this with simple lifestyle measures. Insulin resistance is reversible. However, once you get to pre-diabetes and metabolic syndrome, now you have a 50 to 80% decline in beta cell insulin secretion. You've lost many of those cells. And if the cells are gone, you cannot get them back. So this is more difficult to manage, and you have to try to squeeze as much insulin as you can out of those remaining 30% or so of your beta cells. And then when you get over 80% decline, then you're now in overt type 2 diabetes. So the problem with the US Diabetes Prevention Program and some other programs around the world that are similar is that they tend to focus on screening for people with pre-diabetes. And by that time, a lot of damage has already been done to the beta cells. So our approach is to focus our efforts more on the yellow zone. Talk more about that. All right, so here's our path to type 2 diabetes. So if you look at markers that are used, conventional markers in the clinic to screen for diabetes risk, if you're screening, so conventional markers for diabetes risk include hemoglobin A1C and fasting glucose. So the problem with this is that early metabolic imbalance, that when the first problem is occurring, is invisible to the conventional screening for diabetes risk. If you're screening for cardiovascular risk, you might look more at metabolic syndrome, which has the overlapping definition. It includes glucose, triglycerides, and HDL. All of those are normal as well. So a physician who is screening his or her apparently healthy patient for risk for disease is going to run panels of tests that do not pick up early metabolic imbalance, and unfortunately pick up the damage when it's a little too late. So here's the other part of this EMI, is that it's prevalent in the US population. This is not a minor population. It's close to 10% of the US population, and it's about 20% of teenagers and 15% of young adults. So this is the time to intervene. And unfortunately, it triples the risk for future diabetes. So a 25-year-old with EMI has 3 times the risk of becoming a middle-aged person with type 2 diabetes. And part of our research is to document this at the population level, doing analyses of population data. Okay, so relaxometry to the rescue. So this was a serendipitous discovery on our part. We were using relaxometry primarily to look at non-water components of the blood, especially using T2. But in the course of studying our first group of human subjects, we found that the water T2 actually had even more information in it than what we were looking for. So we switched our gears toward the water T2. And as Istvan mentioned, we published our first discovery phase paper on this in late 2017. We call this the Fort Worth T2 study. It was a study of 72 asymptomatic, generally healthy subjects, ages 23 to 80. And we did a comprehensive health assessment, more than you would get in a clinic visit, with a history and physical plus 130 blood tests and biomarkers were measured on each of those subjects. So why water? I wish we would have thought of this ab initio, but we stumbled across this as we were making measurements. Of course, water is ubiquitous and abundant. It has unique properties. It hydrogen bonds to every protein and lipoprotein in the blood. And the key to this is that shifts in metabolism affect the rotational and translational diffusion of water. And we can pick that up through the T2 measurement. So we like to think of water T2 as the global surveillance system of the blood. I suppose the water molecule looks a little bit like a surveillance camera in the ceiling. So So this is a brief summary of how this works. We can talk more about this later if we wish. Insulin resistance drives water T2 down via alterations in triglyceride-rich lipoproteins. The fact that insulin is not working the way it normally does causes these particles to be remodeled and changed, and the water T2 picks that up. It also picks up inflammation as a drop in T2 by increases in acute phase proteins, inflammatory markers, if you will. It picks up the procoagulation state by increases in fibrinogen. It picks up hypercholesterolemia by increases in the number of LDL particles. It also picks up oxidative stress, also related to the number of LDL particles because they don't get metabolized properly. So that it's picking up multiple things at once going on in the blood, because that's what this syndrome is. So here's an example of some data from the Fort Worth study. This is 72 apparently healthy human subjects. On the y-axis is a marker of insulin sensitivity. The inverse of that is insulin resistance. So the low values mean insulin resistance, the high values mean insulin sensitivity. And here's plasma water T2. There's a very strong positive correlation between insulin sensitivity and plasma water T2. You see the correlation coefficient of 0.64. Keep in mind, in human subject studies, there's so much biological variability that to see a correlation this strong is truly remarkable. And we saw this, actually, we began to see this after 10 patients. So it was really, it stood out. So high levels of insulin resistance lowers the T2. We were able to calibrate a cut point for what constitutes healthy T2 versus a non-healthy T2. And we're able to predict this with 100% sensitivity and 87% specificity. In other words, water T2 is able to detect insulin resistance at those levels, which is also really good for a clinical test. Here's some other correlation coefficients. In addition to insulin and markers of insulin, there's the ApoB, which is a marker of LDL and HDL cholesterol, fibrinogen, white blood cell count, C-reactive protein are all inflammatory markers. You can see those bivariate correlation coefficients are strong as well. When you put this in the multivariable linear regression models, even after adjusting for plasma total protein or viscosity, you still see independent associations with insulin resistance, inflammation and coagulation, and dyslipidemia. And so they're all independent predictors. So this was the end of the discovery phase. So we thought, well, what do we do next? We have to validate this. And one way to validate it is to use a different study in a different population of subjects, and ideally a larger one. And so we picked the PREMIER study, which was a clinical trial done in the early 2000s. It's a multi-site randomized controlled trial looking at lifestyle modification to reduce blood pressure. 810 subjects were randomized into 3 treatment arms. We did an ancillary study on this by acquiring the biobank plasma and serum samples from the National Heart, Lung, and Blood Institute and measuring each one of those samples. So it enabled us to do a larger population, a more racially diverse population, a population with a higher median BMI, so we could look more at obesity and its relationship with T2, and most importantly, a clinical trial of lifestyle interventions, so we could see how T2 responded to the lifestyle changes in these subjects. The interventions were, there are 3 of them called the established plus DASH, and the advice only. The established is a collection of things that were known to lower blood pressure, weight loss, physical activity, reduce sodium and alcohol intake. The addition of the DASH diet involved a low-fat diet rich in fruits, vegetables, and dairy that's similar to the Mediterranean diet. And then the advice-only arm was supposed to be the control arm with a 30-minute session with a dietitian, and the subject was given a brochure and sent home for 6 months to work on their lifestyle modification. So I'm going to show you what the data looks like. So this is the 810 subjects from PREMIER at baseline showing the distribution of plasma water T2. And the first thing you notice is that there's a very wide distribution of T2 values. The higher values are consistent with healthier metabolism and lower risk for diabetes. The lower values are consistent with unhealthy metabolism and higher risk for diabetes. And to make a long story short, we went through a multi-stage process of machine learning algorithms to screen the variables, followed by categorizing them using principal components analysis, followed by multiple linear regression analysis, followed by more machine learning to validate the regression results. And this is the end result of what we saw. In the middle is the plasma water T2, and that's The decline in plasma water T2 or lowering of that is associated with procoagulation, subclinical inflammation, growth factors, especially hepatocyte growth factor increases, lowering of retinol, which is vitamin A, increase in adiposity or BMI, of course, insulin resistance, just like we saw before, poor physical fitness, dyslipidemia. And so all of these things together work together in this early metabolic imbalance syndrome, as well as in metabolic syndrome, to drive plasma water T2 down. So it really provides a global view of cardiometabolic health. So if I was standing across the street from the campus I'm on and somebody asked me, what does the Earth look like? I would say, well, there's some blue skies, some rocky, angular looking mountains, sparse vegetation, some buildings in the foreground, some buildings in the Spanish Renaissance style of architecture. And so that would be my description of the Earth. However, if I was an astronaut and somebody asked me that same question, I would say it looks like this. And so the question is, which one of these is more accurate? And the answer is, they're both accurate. They're just looking at the Earth from different perspectives, a bit like the stationary and rotational frame of reference in NMR. In medicine, what we tend to do is we tend to run tests that are local in nature. They look at a particular metabolite or metabolic pathway, fasting insulin or C-reactive protein, or triglycerides or cholesterol, and so on. These are designed for diagnosis. You have a sick patient, physician is trying to determine what's causing the illness. You need locally focused tests to be able to rule in and rule out different causes of disease and symptoms. However, when you're screening for health status, what you want is something that's global. And T2 is a global measurement. It actually picks up all of these things going on in one measurement. And that's what you want for screening. The doctor's asking, is my patient healthy or not? Because when you're doing screening, you're doing this on people who appear to be healthy. You're not trying to diagnose disease. You're looking for people who have hidden risk for disease and appear to be healthy. To justify doing that, you have to have something that's simple and practical. You need to do this in one measurement, ideally, and that's what T2 is really good for. So the last question we had with Premier is, does the water T2 change with the lifestyle intervention? And the answer is yes. So across the top here, we have 3 columns, the 3 treatment arms of the clinical trial, and then the 3 rows at the bottom are the patient visits, the baseline visit, 6 months after the intervention started, and then 18 months after the intervention began. So we looked at the change in T2 from the 0 month to the 6 month and the 0 month to the 18 month for all 3 treatment arms. And to summarize a lot of data, in treatment arm number 1, we saw T2 increased after 18 months by 19 milliseconds. That was statistically significant. In the established plus DASH treatment arm 2, we saw a similar increase, but the best increase, or at least an equivalent increase, came in treatment arm 3, which was the advice-only control arm. So even 30 minutes with a registered dietitian and a brochure at the end is enough to improve cardiometabolic health if you have motivated subjects like the ones who participate in clinical trials. So this T2 is very good at tracking improvement in metabolic health, not just in screening for it. All right, so we've talked a lot about the results. What about the measurements? How do we do this? You can do this with existing instrumentation. A Brooker MQ20 relaxometer is actually a good device for it. It's overkill. It's far too big and too complicated, I guess, in some ways. More complicated than you need to do this kind of analysis. You can do it with smaller, simpler tabletop devices as well. We have both in the laboratory. We do this at low field, half a Tesla, 20 megahertz for proton, permanent magnet, simple operation, low cost, no shim coils, no cryogens, very simple device. The sample size is also very small. So the Brooker MQ20 comes with a 10 millimeter probe. That's way more sample than we need. In fact, if we use that much sample, we get radiation damping in our results. So we use a much smaller sample size, like a 3 millimeter coaxial insert inside of the 10 millimeter tube, or you can use, if you had an instrument designed with a 3 millimeter probe, you could do it that way. And the idea is to use a capillary size sample. So this is all time domain measurement. We're collecting a CPMG using a time domain CPMG pulse sequence. It's got a few modifications to it, but it's basically just a straight CPMG with a sampling of the signal intensity in the middle of the 2 tau time delay between 180-degree pulses in the pulse train. So we can sample this many times in real time We're not collecting spectra with different CPMG to delay times and then taking an array of spectra, Fourier transforming those and then processing them. We're actually collecting the decay curve in real time. So it's a very efficient experiment in the time domain. And here's our real decay curve. It looks like just a drawn line, but those are actual data points. The signal to noise is so good in this case that you can't even hardly tell there's noise in it. We fit this through a 3-component exponential to tease out the T2s. You can do this with a continuous inverse Laplace transform, but the better way is to do it with a discrete ILT, because that allows you to define the number of exponentials in the fit, and it makes the math simpler and more robust, and we get highly reproducible results. So what we see here is 3 peaks in this component fit, the large blue peak on the right and the small orange peaks on the left. The 3-component fit is shown below for the discrete. So this is the plasma water T2. So the movement of this peak along the x-axis is what we're looking at. And then you can get intensities as well. So the first generation test was plasma or water T2 on 40 microliters of plasma or serum. To do that, you have to do a venous blood draw. What would be simpler and more practical than that? How about using whole blood? So we explored that possibility. And the reason is because in the clinic workflow, if you can avoid venipuncture, then you've really simplified the workflow dramatically. So in order to do this in most primary care clinics, the patient's sent to an offsite clinical lab, the blood is drawn at offsite, analyzed in an offsite clinical lab, and then the results are sent back to the physician in usually a couple of days. This is a slow and rather expensive process because it involves shipping samples and everything else. If you could do it at the point of care, that would be much, much easier and more practical. So we looked to see if we could do this with a finger stick drop of blood using whole blood without having to spin out the cells. And this is what the whole blood T2 looks like. Here's the relaxometry T2 profile. We get now 2 water T2s, the T2 of the supernatant after the blood cells have settled to the bottom of the tube, and a T2 for the pellet. So we call these T2S and T2P for supernatant and pellet. Interestingly, so our goal here was to try to tease out plasma water T2 from the T2 of the supernatant. But when we looked at the correlations, the strongest correlations were seen with hemoglobin, hematocrit, red blood cell count, and blood viscosity. And so what in the world? We're looking at supernatant plasma, which really has no cells in it. So why are we seeing blood cell information? We had to scratch our heads a little bit. So we did an experiment to try to understand this. We took blood out of a subject. The subject in this case happened to be me. And the blood was manipulated outside the body, ex vivo. We diluted the whole blood with its own plasma to decrease the hematocrit, and then removed some of the plasma in order to increase the hematocrit across a series of samples. And this is what it looks like. We did it twice. The gray dots and the black dots are the 2 different experiments. The top curve is T2 of the supernatant. The bottom curve is T2 of the pellet. And you'll notice that the reason we did it this way is because in human beings, if you just look at living blood from human beings, the hematocrit only varies from about 35 to 50%. And what we wanted to do was sample it across a broader range. So that's why we manipulated it outside the body. And you see that T2 of the supernatant has this very steep drop off as you get above about 50% hematocrit, whereas the T2 of the pellet doesn't change at all. So we remembered, of course, that the pellet has hemoglobin in it. And if it's deoxyhemoglobin, that has unpaired electrons and is paramagnetic. So now we have a more paramagnetic pellet with a less magnetic supernatant sitting on top, which creates a magnetic susceptibility gradient, a vertical gradient in the configuration that I showed. And this results in paramagnetic relaxation enhancement, which has a 6th power distance dependence. So the way to think about this is that at a high hematocrit, the water molecules in the supernatant on average are closer to the pellet than they are when you have a much taller plasma supernatant. And so you get this 6th power dependence. So if you simply correct for hemoglobin to the 6th power, you can get back the plasma water T2. Another way of saying that is you can extrapolate back to 0 hematocrit, which is plasma. So that allows us to do all this with whole blood. So first generation was plasma and serum, second generation was whole blood. What's even simpler and more practical than that? How about just looking at the living finger? And in the beginning, we were hoping just to measure blood in the living fingertip, but what we realized after doing this is that the dominant signal comes not from blood, but from the adipose tissue in the finger. But adipose tissue is another window into metabolic health. And so here's a device that we had custom built for us by Resonance Systems in collaboration with Costa Zentar. And so this is a portable magnetic resonance relaxometer designed to measure the distal segment of the living human finger. This is my postdoc with his finger in the device. You put your finger in there for 1.5 minutes and you get a really nice T2 profile. And here it is. Same CPMG pulse sequence. Here's a decay curve from the finger itself. Same discrete ILT. And now we see three peaks here, 3 T2s, if you will, that after looking at a variety of samples correlate with lipid triglyceride T2 profiles. It looks like olive oil. The tip of the finger from the vantage point of this relaxometer, it looks like olive oil. We even put a Slim Jim in there. It looked a lot like that as well, but the peaks tend to shift a little lower for the Slim Jim. So 1 minute and 10 seconds with another 30 seconds for calibrating or setting the field offset. So it's a very simple experiment. So there's a living fingertip. You can see this yellow tissue, which is the adipose tissue, which dominates the fingertip, at least dominates the soft tissue. And we get 3 peaks, which correlate after doing spectroscopy to assess the assignments. The lowest T2 corresponds to the most rigid part of the molecule, the most anchored part of the triacylglycerol, where, especially in the glycerol backbone. The peak way over here represents the methyl end, the omega ends of the fatty acids, especially the ones that are not unsaturated. And then the middle domain corresponds to a lot of hydrogens in the middle of the molecule. And then what we're really looking at here is how this configuration, which is not, it's not really shown as a conformation, but when these molecules pack together, the cis double bonds create kinks which distort the shape of the molecule and limit the packing and make the fluidity higher. So what we're really monitoring is the fluidity of the adipose tissue and the living finger. We've shown this by using lipid model systems to demonstrate how fluidity changes with T2. All right, so to summarize all of this, if I want you to remember one thing from all of this, I would say 3 words: early, in the sense that T2 can screen for early metabolic imbalance to try to preserve beta cells to prevent the slow progression towards type 2 diabetes and cardiovascular disease. It's practical, or it's global. It gives a global view of somebody's health rather than a local focused view like many diagnostic tests do. And it's practical. You can do it in simple tabletop devices with simple samples, including the living human finger. There's a lot of work to do on the finger T2. There's a lot we don't know about that yet, but the blood T2 tests, we have strong, robust data showing that it's clearly related to cardiometabolic health. So with that, I will thank the folks who have worked on this. There are a lot of them. Thank the NIH for funding the premier ancillary study and for 2 institutions that have made this possible, including filing some patents on this. So thank you very much.
Thank you so much, David. This is absolutely fantastic. I keep following this over years and I am getting amazed over and over again. And I wish you luck and success with these devices and then transferring to the real clinical conditions. I wanted to say that questions and comments will be welcome and welcome now as well, although we are running out of time a little bit. Please either identify yourself if you want to have a question, or even better, submit your question or comment in the chat. And this set of talks will be followed by a discussion segment where more informal exchange of ideas and then questions will be possible. And then the presenters will also check the questions or comments submitted and will answer accordingly, and then we shall expect people speaking up in person as well. So currently we only have one. Okay, here is a question. Brendan Dugan asking that does the lipid T2 correlate with EMI as well as the water T2?
Yeah, that's a great question. We are trying to answer that right now by looking at a series of human subjects and making measurements with the finger T2 and also doing extensive metabolic phenotyping to be able to interpret the finger T2 measurement. The answer is the jury is out. We do know that it certainly monitors the fluidity of the lipids in the finger, which in turn reflects the fatty acid composition of the diet. We do know there's epidemiological data that links dietary intake of fats with diabetes and cardiovascular disease. That's well established. The question is whether finger T2 is tracking with that. We will know the answer to that question sometime in the near future, but we have more work to do.
So let's add one more quick question. What is Travis Gregor is asking and expresses gratitude. I really enjoyed it. What is the alternative diagnosis for early metabolic imbalance?
What is the alternative diagnosis?
For EMI, if there is any other method, as I
well, how else can that be diagnosed besides using the T2 method?
So as I was pointing out in the Earth, the picture of the globe on that slide, there's local versus a global perspective. What you could do is you could run a panel of existing tests to get an idea of whether somebody had that. But for screening, that's just too expensive and too impractical.
Yeah.
So yes, we can do it in the clinic by running a panel of tests, but those panel of tests will cost about $450, $500. You'd rather do this with one simple test that costs maybe $20.
Very good. Agreed. Thank you so much. Very, very good.
So I have no intention to cut anything short, but in order to save time, let's leave Don Bouchard's comment and question to the discussion part, which is already a little more complex note. And then let me pass the torch to Aryani. Sorry, I knew I'd do it. Aryani. And then she will be next in the line for presentation.
First of all, I want to thank the panel to invite me for today's session. And excellent talk by Dr. Cistola. I really enjoyed a lot to learn what's happening with NMR. So today, as Istvan said, my name is AaryaniSajja. I'm an assistant professor in the Department of Biomedical Engineering at the University of Memphis. And I'm also a visiting scientist in the Department of Diagnostic Imaging at St. Jude Children's Research Hospital. So if you don't know Memphis, Memphis has the 1.1 gigahertz NMR. So, which is, I think, one of the kind in US. So, I mean, and it is installed at St. Jude, mainly for structural biology. So just to start with, so our lab does MRI and spectroscopy. So to be honest, I'm from MRI background. So I did all my pre-doctoral and doctoral work was on MRI, and then I shifted gears. So as a assistant professor, I started my research in NMR because of very exciting collaborations I formed here at University of Memphis. So, I mean, if you don't know MRI and MR, it's the basic same NMR phenomenon. So it's just here you're acquiring images, but here you're acquiring spectra. So my lab, we are interested, we collaborate heavily with the College of Health Sciences, and we are interested in looking at the dietary effects to find out the biomarkers. So why metabolomics? So metabolomics is more time sensitive compared to other omics, and it will give more real-time information. Of course, it is also susceptible to other environmental and physiological factors, but you come up with a design and you have control parameters, then it would give a lot more information, which could be used as biomarkers for disease. So today's talk, I'll be presenting this paper that we recently published in July 2022. So it's multi-tissue time domain NMR metabolomics, which is investigation of time-restricted feeding in male and female Nile grass rats. So just to summarize, the main purpose of this study is to use time domain NMR to do multi-tissue metabolomics, because we want to do a systematic approach, and then to quantify the metabolites and see the dietary differences because of high-fat diet versus time-restricted feeding. I'll tell about a study, experimental study in a minute. So I know all the audience here know conventional Fourier transform NMR, so which is actually used for quantifying the metabolites. But as you all know, it involves a lot of pre-processing the spectra before quantifying it. So it has zero filling, epidization, phase correction, and baseline correction, which all has to be done by a user. And it is very user dependent. And finally, we quantify the metabolites using integration and peak fitting. So integration is, again, you're quantifying the area under the curve, which is reflective of the concentration of the metabolite in the sample. And peak fitting is where an expert would fit the peak and, again, calculate the area. But again, this is very subjective. So there's a lot of manual input here. So this would introduce a lot of bias and affect the accuracy of your measurements. So our lab was interested in moving more towards automation. So automating the metabolite quantification from the NMR spectra. So as you know, so this is the FID on the left side, showing the free induction decay, which is a time domain signal, and you do Fourier transform, you get the spectra. So these both are related, right? So you have this amplitude here, which is nothing but that is reflective of the area under the curve, and that's used for the molecular or metabolite quantification. Similarly, the period here is reflective of the frequency, and the decay rate corresponds to the line width. So, I mean, I'm going to talk about time domain NMR, which was inspired by Dr. Krishnamurthy, who is here on the panel. So we came across the Kraft NMR, which he published in 2013, which kind of converts all your data and produces like a table in the form of a table. So you would get like amplitude of all your peaks, and then you can quantify you can determine the concentrations of the metabolites. So these are other papers that he published. And it's also, the Kraft is also used for the 2D NMR data, but today I'm going to only go with the 1D NMR data. And I recommend, I mean, this is an excellent review paper that was published, which shows the application of time domain Kraft for 1D, benchtop, 2D, and so on. So if you haven't read, this is a very good article. So I would just explain Kraft in a nutshell. Again, you have the one that is who founded Kraft here, Dr. Krishnamurthy. So you can ask more technical questions for him. But so you have the original FID, and what you're doing is, this is your spectra. So you select the ROIs from here, and then you do digital filtering, which would give sub-FIDs. And then you do the Bayesian decimation and convert these into amplitudes. And in our case, for the metabolomics, we also have a quantitative reference. So we can also calculate the concentrations of the metabolites in the sample. And it also produces visual output. So how sure you're with these numbers, then you can see the visual output here. So for example, this is showing the experimental spectra and the Kraft spectra and showing the residues. So which you can see, which helps in knowing if there is any errors in the quantification. So we were, as I said, inspired by seeing that this could increase reproducibility and automation and decrease the operator bias. Because as I said, if you have to do in the frequency domain, you have to be consistent, and it's more susceptible to errors. So our main study objectives were, first, we want to validate the Kraft approach using complex mixtures and metabolites, so which I'll tell how we did it. But first, we want to have the fingerprints first in complex mixtures, and then quantify them, and then compare it to the volumetric measurements to make sure that we are quantifying right with this technique. And then we use this approach for multi-tissue metabolomics. So this is actually the journal cover image that I put here, but I'm going to talk about the study in my next slides. So this is actually our first paper where we used Kraft for NMR lipidomics. So what we did, again, we used, we opened the FID and we selected the fingerprints. So first, as I said, we wanted to validate in mixtures. First, we have used mixtures and then assigned the ROIs and then quantified first the amplitudes and then the concentration using the internal reference standard. So these are showing the list of lipid metabolites that we quantified. So as I said, so first we compared in this paper both Kraft with the Fourier transform NMR. So this is comparison with the ground truth or volumetric concentration to make sure we are getting good correlation between the ground truth and our Kraft measurements. So we got good correlation coefficients and the slopes were close to 1. And again, this is for the lipid standard. So we prepared different mixtures with known standards. And then we also did the same for aqueous standards. So we prepared mixtures and verified the concentration using the Kraft and with the volumetric concentrations, and it produced very good correlation. Again, this is just in the form of table comparing Kraft with the Fourier transform NMR, which is the standard way of quantification using deconvolution. And then after we defined our ROIs in the mixtures, we applied the same ROIs in the tissues now. So we got all our, these are showing the spectra in complex mixtures, and then we applied those ROIs in the tissues in the study, which I'm going to talk. So right now, so far, I have told how we defined our ROIs in the standards. And as Dr. Cistola mentioned, so metabolic syndrome is becoming more prevalent in the United States. I mean, all over the world, especially in the United States, about 35% of the US population has metabolic syndrome, is affected with metabolic syndrome. Again, this is characterized by obesity, insulin resistance, hypertension, lipidemia, and so on. So our main interest is to investigate high-fat time-restricted feeding. So studies have shown that time-restricted feeding can be effective in weight loss strategy and improve the lipid profiles. So we were interested in seeing if even though the subjects were given high-fat, but if it is given for certain time each day, would it bring back their profile to the normal profile? So that's the main intention of our study. So what we did here is we had 4 experimental groups. So the first experimental group was fed with chow diet, which is a standard diet. And we have 3 more experimental groups. So the second one is fed with high-fat diet ad libitum. And we have 2 more groups which are fed a high-fat diet, but only for the morning 6 hours. So from 8 to 12, they were fed. So this is in relation to the circadian rhythm. And these were fed in the evening from 2 to 8. So we wanted to also see whether morning versus evening, would it cause any difference or would it affect the lipid profiles? And we chose Nile grass rats for this study. Again, the usual animal model is mice, but we went to the Nile grass rats because it's a diurnal model and it would develop a constellation of metabolic syndrome symptoms similar to humans. So again, this is more new recent model, but we wanted to see the effects as it resembles the human diurnal model. And we also looked into sex effects because males and females have differences in metabolic, metabolic task, how they do. So here we have, we tested both male and female Nile grass rats, and we followed those diets for 6 weeks, and then we harvested the tissues, and we looked into liver, heart, and adipose tissues, which are the metabolic organs, important metabolic organs. And then we extracted the metabolites, measured them using NMR, and then processed using time domain Kraft using the same fingerprints we used in our standards, just to maintain the consistency so that there is no user bias. And these were scanned on a 400 megahertz NMR. So this is just showing how we extracted. So we extracted the lipid and aqueous parts. So we used MTBE extraction, for that. So after extraction, we dried out the samples using nitrogen, and we added solvents and reference standard, and finally scanned them using NMR. So this is showing the Kraft fingerprints we used. Again, this is only for the lipids, but it's showing that across the tissue, we applied the same fingerprints again to make sure the results are consistent. And DMSO2 was the internal standard that was used for the lipids. And here, I know these are all short forms. The TG means the triglycerides, and you can read from here. PC is the phosphatidylcholine. LA is linoleic acid, DHA, monounsaturated fatty acids, omega-3, total cholesterol, and total fatty acids. And so this is our first result showing the two-way ANOVA that we did in chow versus high-fat diet, ad libidem diet. So again, it's we measured across liver, heart, and adipose. And the circle one are the one that show significant differences in lipids here. So all these like triglycerides, total fatty acids, those were different between, those were higher in high-fat ad libidem diet versus compared to chow diet. And we saw both the significant differences in the liver and heart, and also for some lipids in adipose tissue. And we also saw decrease in the aqueous metabolites in high-fat ad libidem compared to the chow diet. So the lipids were increased and the aqueous were decreased in high-fat ad libidem diet compared to the chow diet. And then this is showing the time-restricted feeding versus the high-fat ad libidem. So it's showing both the AM and PM. So AM, again, is the morning fed rats, and PM is the evening fed rats. And here also it's the circles here are showing the significant differences. So it's showing that in these for these lipids showed decreased levels compared to the high-fat ad libidem diet. So the time-restricted feeding groups showed decreased lipids compared to the high-fat ad libidem diet. So we saw again both in liver and heart, but nothing in adipose. So just we broke up into two parts. So first one, with respect to the high-fat diet phenotype, we saw changes in fatty acid profile in case of adipose, but we didn't, so the saturated fatty acid to unsaturated ratio increased. The saturated fatty acid percentage, monounsaturated fatty acids and percentage, and there's decreased omega-3 fatty acids. Whereas in liver, so this is for the adipose, in liver we observed there's ectopic fat accumulation. Again, it's evidenced by increased lipid amounts, as I showed in the previous slide. So all these fatty acids, triglycerides, and total cholesterol were increased compared to the standard chow group. And there was decreased in aqueous metabolites. Again, these are amino acids, so there should be some disruption in the amino acid metabolism. That's why we see a decrease in aqueous metabolites in high-fat diet. And we saw a change in fatty acid profile. So if you see here, now you see increased monounsaturated fatty acids, whereas decreased polyunsaturated. So again, increased saturated means it's going towards the bad direction. And so compared to all the three tissues, we saw highest percentage of significantly changed metabolites in the liver. Again, liver is the primary metabolic organ, so most of the differences we saw are in the liver. In heart, we saw, again, fat accumulation evidenced by some increase in the lipid amounts, and also change in the fatty acids with the increased monounsaturated fatty acid percentage and decreased polyunsaturated acid percentage. So that's for the high-fat diet compared to the chow diet. Whereas for the time-restricted feeding phenotype, what we saw in adipose, we didn't see any differences. There were no detected metabolic changes. Whereas for the liver, we saw that in TRF, they were lower. So all these lipid levels were lower, whereas it showed higher acetate, which is a aqueous metabolite. And again, liver showed the highest number of increased, highest number of metabolite that showed significant changes. And in heart, we again saw lowering of total fatty acids and saturated and monounsaturated fatty acids compared to the high-fat ad libidem diet. And surprisingly, we also saw, again, males and females, it's evident that they manage the metabolic task differently. So if you see, this is a male liver, it's more red, and female, it's more yellow. So we also looked into biopsy samples. So if you see here, again, males, so all these white circles here are fat deposits. So if you see here, males, you see less amount of fat, but females, you see something is going on. So it's storing fat more in the livers. Females are storing fat more in the livers. And we also compared this, so the liver weight. So if you see here, again, the females with a high fat diet, the liver mass is more compared to males. Whereas this is adipose tissue, ectopic adipose tissue, but here we see the opposite. Here, males, it was high in high-fat ad libidem compared to the females. So it means that, again, females somehow are storing more fat in the livers, whereas males more in the ectopic adipose tissue. So that's the observations from the metabolic functional parameters, but from the NMR lipidomics, again, the liver for the females showed higher lipid levels compared to the males. So again, all these were higher in females, and aqueous metabolite levels, whereas were higher in males compared to females. So we saw the opposite. And in heart, for the heart, we saw increased aspartate in females compared to males, and in the adipose, we really didn't see any differences between males and females. So this part of the study was actually published as a separate paper, which was focusing more on the functional parameters. All right, I think that's all I have. So the final study conclusion. So we used Craft again, which helped to automate our processing and make it more systematic in measuring all these metabolites accurately. And TRF was protective against some effects of high-fat diet. So even though they are fed with high-fat diet, but we saw that the TRF has decreased the lipid levels in those tissues. And as we saw, more changes are evident in the liver metabolome. So it was liver metabolome was more sensitive to high-fat diet. The other tissues also showed some response, but liver was more sensitive. And also we saw more sensitive metabolome for the liver in case of the time-restricted feeding compared to heart and adipose. And we also saw differences between male and female, mostly in the liver metabolome. So with that, I would thank my lab. So Hayden Johnson, he was the first author on this paper, and he's my first graduate student, first PhD graduate student in my lab, and he's the main contributor for the study, and have two more undergraduate students who contributed. So Thomas and Gary, they did extractions for the heart and adipose samples. And I would like to thank my collaborators from College of Health Sciences who helped us with all this, running the mice samples and everything. And I would like to acknowledge my funding sources, NSF support for the Joule spectrometer in our chemistry department, and also for the NIH Trailblazer. This is more for the MRI research. And I would like to thank all the members of my lab. It's a wonderful lab. And with that, I can take any questions.
Thank you very much, Aryani. This is a very thorough and carefully conducted research, very extensive and beautiful. I leave others for questions. Let me just make one comment. that it seems that now it is clear that either a big lunch or a big dinner is a good one, not both.
Yes.
As far as fat is concerned. Maybe some red wine helps to dissolve it. All right, so let's just have one question if somebody wants to present it in the questions or chat. But given the time constraints, it would be perhaps better to leave it for the discussion period. So here is my presentation, at least where I start. And before I go to our own part, there is something I wanted to share with you very quickly, because Emmanuel Hatzakis couldn't make it, unfortunately, but there is a fantastic new paper out published in 2023, which is very much lined up to what Aryani was talking about, combining or comparing Kraft with FT-NMR. Now, this paper is a direct comparison, and in that sense, it is highly technical. And it is kind of a groundbreaking comparison for studying metabolic mixtures. This is the reference, what you can see here. And I got permission from Emmanuel to share some things with you from the paper. Let me see. That's how I should go. Yes. This is one figure out of this paper, which compares Kraft and conventional Fourier transform NMR analysis. And you can see that there is a clear difference between the two. And again, I just don't have time to go through the details, but here is the overall conclusion, which I share with you, and then it will be recorded as well. The conclusion is that Kraft is very competitive for such analysis. For PCA, it is perhaps even better. For supervised statistics, it is directly comparable, but there are notes in the paper that time and the simplicity of the analysis makes it really competitive and perhaps better. But there will be more analysis required for clarifying all details. So with that, I go to our own presentations, where there is an importance of sex separation. So this is when the stars line up very nicely suddenly, because both of these presentations, which you heard already, point to the things which we ran into. The sex separation is not widely acknowledged, but Aryani showed exactly how important it can be, and I have some more notes about it later. These are my co-workers at the first level, although there are many other students who are involved. Professor Ortved and Sarah Ralston are the major collaborators for long years. Ken Conover is my colleague and friend who is contributed to many of these projects. John Spears and Dan Kim presented senior theses, which are the foundation of this following presentation. And Michelle Coleman provided samples from Texas early on. Now, this is another acknowledgement of my wonderful group from this summer, all undergrads from 2 summer programs, Blake, Jennifer, Jackie, Patty, and Sage. And there is a paper, because they did such a nice job, where they all will be authors for the Journal of Chemical Education. So cutting into the chase, FT-NMR and TD-NMR, in this case, relaxometry. Of course, here compares like this. This is a conventional spectrum, which provides details and quantitative spectral information. The relaxometry, this is one representative spectrum, provides very limited information in terms of number of peaks or numbers, but the position of the water and also the position and the composition of lipid peaks, in this case biofluids, is informative in comparison what is going on with the different samples, especially in large number of comparisons. We often study osteochondrosis dissecans, which is one of those inflammatory diseases of the hooves of the horses, also laminitis, and it is related to obesity and diabetic conditions. Now, this is I run through very quickly, but this is something I overheard by somebody, that genome is the letters, proteome is the words, and metabolome is the story. I love this. I keep telling everybody, students included. There is some history about the metabolomics and metabonomics as the historic terminology. These are the major methods we use: multivariate statistics, S-toxy, and TD-NMR for our studies. This is a typical sample preparation. We insert a tube with the sample, and there is D2O for lock and the shimming in the outer between the walls. And we have 500 and 800 megahertz cryoprobe equipped systems to run experiments. Now, again, coming back to this typical spectrum, this is a full spectrum where the water is suppressed, but other than that, we visualize everything what is in the sample quantitatively. What is most important in this aspect, that the large molecular weight species such as lipids, lipoproteins remain all visible and participate in the spectrum. That's very important, and so often it is ignored or deliberately removed chemically or mechanically from the samples, because the relaxation filtered or purified samples are much easier to handle, because you focus on the small molecules, which are the metabolites. But lately it turned out over the last good decade or so that these large molecules, which are only represented in very small residual quantities, are very important messengers about the overall metabolism in the system. Now, this is a typical overlay of the spectra, some 40. These are the major multivariate approaches we use, and it is handled in Simca for statistics. Oops, that's too fast. So this is a typical back-calculated coefficient plot for the OPLS, from the OPLS-DA analysis. It is done by one of the students, Patty, in the summer, and that provides you information about the most important metabolites which make a difference between the cohorts of diseased and healthy. This summer program was very successful. Sage Kortsen pioneered a study on her own where she found a correlation between choline metabolism and insulin resistance using S-toxy analysis, and then went into the deep details of the metabolic pathways. Now, this is the TD-NMR, although it has been presented enough times before, so I go through it very quickly. But the point is that we have a lipoprotein proteome large molecule system, which is hydrated with water, and that water exchanges with the bulk water. So we see the weighted average of the two. Therefore, when we see the time domain signal in the TD-NMR, then it gives you information, indirect information about the composition of this big molecular system. It is a multi-exponential decay, and then with appropriate processing, one can analyze the individual components. This is what David already showed. We had a MQ10, which is our relaxometry device. Now, this is what we also add to the procedure. This is a healthy and laminitis comparison for the relaxometry. And you can see that there is significant difference between the two, although there is a lot of variation as well. So it is clear that the lipoprotein or large molecule composition is quite diverse in the data set. But we also do add some additional post-processing in MNOVA, which is appropriate apodization and the zero filling, so the analysis becomes much easier for software and also for individual user. Now, this is a typical example when the healthy and the OCD cohort, it is the OCD study, shows the difference is quite clear, but it is also clear that there is a lot of diversity in both of these things. Interestingly enough, the disease condition seems to collapse the diversity a little bit. including the lipids, although we focus on the water because the lipids didn't give us consistent information. There is something which we are still scratching our heads, because there are several samples, not only one or two, several samples where we see 2 water peaks. And if I apply a strong resolution enhancement, you can see that there are subcomponents within the individual peaks. So my hypothesis or current speculation is that there are lipid vesicles in the blood samples, and maybe in this case, or those cases where we see 2 signals, there are 2 different lipid vesicles or a group of those in the samples. That's even more fascinating, that we may see component resolution underneath. So this is still in the works, and I'll be happy to receive or accept comments and suggestions how to explain this behavior. Now, here is where we ran into the trouble when we did analysis of an OCD data set, and this is John Spear's senior thesis. When we looked at the relaxation filtered experiment, it lined up very nicely in the OP-LSDA. However, when he tried to analyze the full spectrum, which includes the unsuppressed large molecules, lipoproteins, there was a consistent failure where some of the sick samples migrated into the healthy group, and he just couldn't get any better results no matter how he approached it. Now, this immediately highlights that looking at the small molecule subset only could be very much misleading, because this is a real effect. And we received a hint from the TD-NMR analysis, which was done on the same sample set. And here it is, the box plot of it. Just let's focus on the healthy female, OCD female, healthy male, OCD male comparison. If you look at it, the healthy and the diseased for the female has an opposite direction in the metabolic shift than the male. So in a way, the metabolic differences kind of cancel out between the 2 sexes if you don't separate them. So when we did separate the sexes, this is how beautiful the OP-LSDA becomes. And this is only just the fillies and the colts. Sorry for the typo here. So fillies and the colts only for the full spectra. So they are nicely separated and clean. There is one interesting occasion when we did find one sample which was diagnosed as sick, but it just never made it in the sick side with good validation, and it migrated to the healthy. So when we classified it as healthy, then the scores plot became very nice and clean. Such a misdiagnosis happens here and there. We had a publication earlier on when we identified such a thing as well on another sample set. This is Blake's result, which proves also and demonstrates how the sexes are important and separate the clusters. When the OP-LSDA is done on all the 4 clusters, but identifying the classes accordingly, then you can see that the female and male clusters are separate on this plot as well. This is S-toxi results of Jackie, wonderful analysis. And we focused on and keep focusing on glyc-A and glyc-B resonances, which are very visible in the horse samples as well. They turn out to be important reporters of inflammatory conditions. And you can see that the female and the male are quite different, and not only between the female-male comparison, but also all the quadrants are different. Therefore, it is very clear that the sexes have very different metabolic conditions to begin with, even in the healthy state. Consider or look at the lipids. They are. I love that the resolution in the statistics is better than the NMR resolution, because the significance of different subcomponents of the lipids show with the different coloring. And then when it comes to the disease state, it is very clear that especially the lipid resonances, lipid composition has a very different response. Now, what is the biochemistry behind it? That's the next step to clarify, and we are working on it, although this is the stage of the least expertise we have at the time. But there is so much to do more. And here it is. Thank you very much. And now it is time to turn to the discussion phase.
Istvan, this is Krish.
Yes.
I was just excited to some extent to see the result that you showed about the water. You saw 2 water peak.
Yes.
In there.
And it is not only one sample, so it is not an artifact. It happens.
I concur with that. We have seen something like that many years ago, about 15 years ago, when you were looking at, I think if there are some Lilly colleagues in the audience, they might probably remember. We've seen something very similar to that when we were looking at cartilages in vivo, actually it's ex vivo, but MRS, magnetic resonance spectroscopy, localized spectroscopy, and we were looking at that to study the water in cartilage. And we saw some heterogeneity of the proton resonance of the water. And we actually saw that very similar behavior when you take the voxel instead of the cartilage, knee cartilage, if you go to the backbone disc and actually take voxels from the annular region to the middle of the disc, which is, I forget the name, nomenclature for that, where there is more looser backbone structure, you can actually see the water resonance move as your voxel moves from annular region to the middle region. And we see the similar thing in the in the knee cartilage, where in a control knee, where we expect the cartilage to be more uniform structure of the collagen and all that stuff, whatever that structure is, we see a single water frequency, which is different from the bulk water frequency. But when the osteoarthritis sets in, or induced osteoarthritis into the knee, you actually see a heterogeneous water frequency. So you see as much as 4, depending upon how heterogeneous your knee is, you see osteoarthritis is, you see that significant heterogeneity in the water frequency as well. At that time, we actually interpreted that, and we actually did some study as well that we are convinced it's actually the residual CSA, chemical shift anisotropy, residual CSA of water in tissues. They are not as freely moving around, or as you would see in bulk water. And so when the heterogeneity of your tissue changes, the water frequency also changes with that. And we see this, and we actually did one of my colleagues actually did some interesting experiments of rotating the sample itself, and then see as the sample rotates, because you're coming to a magic angle, and you can see the water frequency going back and forth. And so it was very interesting. We never published it. We never really proved anything at that time, but it just brings back memories of that observation we had of water frequency splitting in tissues, particularly the tissue is not a homogeneous tissue.
This is very interesting, and may give us some hint and way to think about it. But the problem is that the serum sample is isotropic.
Yeah, I understand.
At least visibly. It may very well be that these vesicles exist and they are not visible as such. And just taking the spectrum or the FT-NMR doesn't show distribution. But maybe the TD-NMR is the way to identify the heterogeneity of the vesicles.
So if I might pipe in here, so the usual source of heterogeneity in the water like that would be looking at intracellular versus extracellular water. You wouldn't expect to have that in serum unless perhaps some of the samples might be contaminated with platelets, which are hard to see and hard to remove sometimes. So that's one possibility to look at, but I would expect that if that was the case, that second contribution would be relatively minor. And certainly in tissues like what Krish was talking about, you might see some differences between water inside the cell and water outside, because the cells have ferritin, which is a protein that binds a lot of iron to sequester it from the intracellular contents and milieu to prevent oxidation. And the ferritin can certainly change the features of the water inside of cells, unlike what you would see in the plasma or serum.
That's a very good point. Thanks, David. What we should do, and we haven't done yet, that we have to carefully compare the TD-NMR spectra with the FT-NMR, because if there is some contamination with platelets, it should show up in the FT-NMR spectrum as well, one way or another.
Yeah, it's a good idea with anticoagulated blood to spin it a second time to remove any residual platelets. You could use light scattering or some other simple method to determine whether you've got the platelets out, but we take care in doing that when we are analyzing blood.
Thank you. This is a good point. So, who will be next in chat? There is Amy Freund from Brooker. Curious that both animal studies show differences between male and female subjects. Question for David Cistola. Do you see differences in water T2 between male and female subjects? Perhaps T2 cutoffs would be different.
Great question. We do study gender, sex differences to look for that. We control for that in our regression analyses, and we do not see a systematic difference between male and female in humans when it comes to blood T2 or plasma and water T2. It's not a significant variable in our analyses. So I would mention, though, that this phenomenon of seeing male versus female differences, especially in liver fat content, this has been known for a long time, not so much through magnetic resonance, but through other methods. And back in the 80s, I remember some studies looking at that and attributing some of that possibly to the differences in lipid binding proteins inside of liver cells. Liver has a fatty acid binding protein that's highly abundant, and those levels are different in male versus female rats and mice. And there was thought back then that may have something to do with it. But it's really nice to see from Ariana's work that you can pick this up by magnetic resonance.
Until somebody else gets courage to ask a question, I have one, because when you start or look at a finger, does it make a difference if you look at people who work hard with hands, such as agricultural participants, or those who are typing only on computers? And then their fingers are softer, maybe, or what?
We haven't done that experiment, but I think you're talking about how calloused the skin may be.
Yes.
In the fingertip. We're not really measuring primarily skin. We're measuring the underlying adipose tissue inside the finger pulp. And so that really dominates the signals. I doubt that in our measurement that we would see such differences. We also know that we don't see differences, short-term differences in metabolism. So the finger T2 is reflecting long-term dietary intake of different lipids. Ariana alluded to that in some of her work about the composition of adipose tissue. If you, for example, somebody who eats a lot of fish is likely to have a higher finger T2, especially the third peak on the right of our profile, the highest T2 we measure. We're likely to see higher T2s in people who have a higher intake of EPA and DHA, which have 5 and 6 cis double bonds each, and which causes an increase in fluidity of the lipids. We've done that on a simulated scale with model lipid systems and mixtures of lipids that mimic adipose tissue. But we don't see short-term changes. So if you had a lot of, if you ate a big cheeseburger the night before and you came in and had your finger T2 measured, it wouldn't change in the morning. We're looking at long-term dietary habits rather than short-term ones. That's the way the adipose tissue is remodeled.
Thank you. It is amazing with such a simple measurement, you can get so much and extensive information. It is outstanding.
It continues to surprise us.
Mm-hmm. And unlike FT-NMR, where fish and chips immediately shows up.
So as a former protein spectroscopist working at high field, in complicated experiments and pulse sequence and trying to tease out atomic resolution and complexes, we used to try to get rid of the water, of course, try to suppress the water, and now the water is our best friend.
Mm-hmm. So let me invite everybody to present questions or comments. Either you can unmute yourself and speak up or send a note to the chat.
I have one more question / comment, if I may. Now that I'm seeing that there is a silence here. So I saw your, Istvan, it was in your spectra, one of the things that you showed about these lipid blob riding on top of your peaks. And I think this was one of the common problem people face when studying biofluids, this broad peaks that, and then it obviously one of the conventional wisdom is to remove that by CPMG type of experiment or with the current technologies of craft and that kind of things, removing, not bothered by these background. But one can envision the inverse as well. Yeah, that in a computational world, one can envision inverse as well. That is removing all the sharp resonances out of the spectrum and study the spectrum of the broad lipid spectra by computational, because when you do I am borrowing the word TD-NMR here, but TD-NMR processing like Kraft Bayesian method, they are very, very powerful in sharp resonances, meaning those that decay slow. They are very powerful and we applied that significantly in that as well. But you can take that table and subtract out of your FID and have a spectrum or a FID of your broad resonances that you can potentially study either in terms of how the shape of the spectrum look like, or some kind of a principal component analysis of the individual data points. This comes to mind brings to mind about studying things like polymers, backbones of polymers, studying antibodies, medium-sized proteins in the antibody spectrum, instead of worrying about these broad peaks that are in the background. And these are the kind of stuff that one could do with TD-NMR, and I am just commenting that we can do an inverse, quote-unquote, inverse CPMG to study the broad resonances as well. How we will study is, I will leave it up to the audience, but there's an opportunity to take out these slow decaying sharp resonances out of your spectrum from a computational side, and then just study the FID of the slow, of the fast decay.
I would love to see such an analysis, and I have a data set which we could play with. In fact, what you are proposing is the computational version of diffusion filtering.
Exactly.
Which is quite tedious sometimes to suppress the sharp peaks well enough and keep the rest in good condition. And it turns out that there are peaks which are very much matter of interest. Jeremy Nicholson published nice papers about highlighting the glyc A, glyc B peaks and the SPC peaks and removing everybody else. But keeping these guys, we try to look at it just looking at the original diffusion filtered experiment. But again, it is extra work and then not that easy necessarily. So if you can do it computationally from the full spectrum, that is the gold mine, so to speak, for such analysis, and very simple. Computers are getting less and less expensive and more cooperative, so that would be fantastic to check out these capabilities.
And they work 24/7 without complaint.
Oh, that's true. Except if there is a power outage.
There's a power outage. Yeah, we can take it offline with you, and then I can show you some examples.
I'll get back to you sometime about it. Yes. Now there are 3 questions, so why not you speak up? Let's start with Michal Kalinyak. If I pronounce the name well, unmute yourself and you are welcome to present your question in person.
Yeah, hello everybody. I
Hello.
wanted to ask about the ranges for different metabolic disorders, for ranges of T2 for different metabolic stages or disorders or EMI, as well as going to the diabetes field. What are the cutoff values, I would say? Thanks.
So I can speak in general terms about this. In the study that we did, we determined a cut point of 745 milliseconds for plasma and about 745 for plasma for insulin resistance and early metabolic imbalance. And that number is going to change depending on how you collect the data. So it has to be standardized to the particular protocol that's used. So for example, the interpulse delay in the CPMG scheme is going to make a big difference. These are devices that don't have shim coils, so you have a somewhat inhomogeneous magnetic field. So we use a pretty short interpulse delay to minimize diffusion-related changes in T2, but we do get some of that. And so depending on how you do the experiment, it's going to determine what the number is. But if you standardize the protocol, and you do it the way that we do it, then it's in the 750 range. So above 750 is metabolically healthy, below 750 is metabolically less healthy or unhealthy. And the farther you get from that cut point, the more healthy or unhealthy you are by this measure. Cut points are optimized for distinguishing sick versus not sick or healthy versus unhealthy in this case. But they're not perfect. So you have to use a cut point as a guideline.
Thanks, David. I invite Ari. Oh, sorry, I have no intention to interrupt. Michal, do you have more to say?
No, I just wanted to thank you.
Okay, so let me invite Erin Campbell to speak up. Unmute yourself and present your question.
I was just, so we changed it into the discrete inverse. What is the significance of that over the continuous inverse Laplace transform?
So when we first started doing this, we used the continuous inverse Laplace transform algorithm. And the continuous algorithm tries to achieve a distribution of T2s that's consistent with the T2 decay curve. And it's mathematically more challenging. And if you don't have good signal to noise, it becomes an ill-posed mathematical problem. So you have to try, first of all, to make sure you have good signal to noise. But then secondly, you'd like to reduce and simplify the math as much as possible. Some of that comes down to being able to minimize the number of exponentials that you're fitting to. So in the discrete algorithm, you're able to control, since you're not trying to sample the full continuous distribution of T2s, but rather looking at discrete values, you control the number of exponential terms, you simplify the math, and you make it more reproducible. The continuous algorithm is good. It allows you to do visualization, but for quantitative purposes, we use the discrete algorithm as the preferred method. And so if you're comparing, let's say you're comparing 100 different people, serum from 100 different people, you want to make sure you're comparing apples to apples to apples when you're doing the analysis. The discrete algorithm allows you to say, hey, there's supposed to be 3 components that we're resolving in human serum, and we're going to do that on every single subject, rather than the continuous algorithm, which might choose 4 in one run, and 2 in another, and 3 in another, and you're mixing incompatible data sets that way.
I have to apologize to Don Bouchard because I skipped his question because it slid out of the little window I have here. So
Can I interrupt? And I just wanted to continue that discussion from Aaron and
sure.
David. There is an interesting possibility here is also to look at in some ways, say TD-NMR meet TD-NMR analysis, meaning we can look at that. We have been looking at that recently with Bayesian probability function. So because Bayesian has been extremely powerful in fitting these kind of exponential decays, and we have been looking at that, and we have very promising results without needing to do any. One of the benefit of that, there is no precondition in how many, nor is there what function it is for Bayesian. We simply say this is a multi-exponential. Go find as many as you want to fit this, and so it has been extremely powerful. Hopefully, we will have something to talk about soon. I'm just saying that there's a potential opportunity to analyze these things in a different probability-based functions as well.
Yeah, good comment, Krish. There's a number of ways to do multi-exponential analysis, and that may turn out to be one of the better ways of doing it. And there's different kinds of, Laplace transform, inverse Laplace transform algorithms are just one kind of math for doing this. There are other kinds of math. But then within the ILT family, there's different algorithms as well that have differing abilities to tease out information. So there's many different ways of doing this. It'd be interesting to see if the Bayesian approach might be better. But certainly the applications that we're using, the ILT works just fine. But for some other applications, it might not.
We can talk offline on that. Sure. I would be interested to talk more with you about that.
So then let me turn back to Don Bouchard, and with my apologies.
Thanks, Istvan. Good morning. My question is for Dr. Cistola and even Istvan. Measuring T2 at a fixed field gives a trend at a single water correlation time. And certainly there are many interactions of water in complex media like blood fluid and other fluids and other media. My question is whether you think multi-field studies using fast field cycling to measure water dynamics would provide a wider profile of information.
So I'm somewhat familiar with that approach, and we've considered using that approach ourselves. It comes down to what kind of scientific question you're asking, and also what kind of setting you're working in. You certainly will get more information by varying the field and looking at changes in relaxation across different magnetic field strengths. In this case, it doesn't. It comes down to balancing, doing something that's practical enough and simple enough to penetrate the clinic environment versus something that's more scientifically rich and interesting. So depending on what you're doing, you would choose one over the other. So we have not really delved into that, but I watch that field a lot, and I'm very intrigued by what might be done with that in the right setting. The medical field is tough. The doctors are, they're scientists in training, but really their job is to figure out what they can do to help their patient. And so when you introduce some kind of a new approach and you try to penetrate the clinic, the first thing they're going to ask is, how does this help me manage my patient? No matter how scientifically interesting it might be, how does it help me manage my patient? And on top of that, how is it better than things we already have? And so, when I put my doctor hat on, I kind of have to think about it that way. And when I put my scientific hat on, I want to try to make things more powerful and more information rich. And so I have to balance those two types of priorities when we do our work. But fast field cycling is a fascinating technology, and I think some interesting things could be learned from blood samples and other related samples using this method. We just haven't had the opportunity to delve into that.
we should go to the next question. Diana Sandoval.
Hi, good morning. Thank you. So this question is directly for Dr. Cistola. I was wondering, what are some of the benefits of using water T2 rather than the usual gold standard blood tests like hemoglobin A1C, CMP?
Yeah, excellent question. So certainly we're not going to be using plasma water T2 or whole blood T2 to diagnose diabetes or pre-diabetes because there are already excellent tests for doing that. What we're looking at is for hidden conditions that are missed by conventional clinical testing. So hemoglobin A1C is going to pick up changes in glucose metabolism as you move into pre-diabetes and diabetes, but it doesn't pick up early metabolic imbalance. And that's our goal, is to be able to try to screen for that early condition to preserve pancreatic beta cells. So T2 can do some things that conventional clinical testing cannot do.
Thank you, Dr. Cistola.
I just find it interesting that it went out as soon as she went upstairs.
John, it's a hard button to push, and I've turned it on twice now.
Okay, thank you.
That was not a question, I guess.
I have a question for Aryani. One of the banes of metabolomics is the peak position not staying where they are, and alignment has been aligning spectra, multiple spectra from different sources, has been one of the big challenge in the metabolomics. Did you do any particular alignment tool, or did you find your selection of your fingerprints or within the limits of the peak position movement?
Yeah, I mean, yes, we had to do some moving, but I think we didn't change any width or anything.
Okay.
And I could poll my student. He should be in here. So test question for him. Hayden, are you there?
Yes. So sometimes, like for the most part with these lipids, there was no need to move any ROI.
Okay.
It was just consistently in the right spot, but I did double check visually before running crafts just to make sure. There was a few times where maybe something changes in relative amplitude or something, and maybe I need to scoot something the tiniest bit to the left. But for this data, it was not a problem for lipids. Now, if it was a problem, yeah, I would think about using maybe a spectral alignment tool like ICO Shift or something. But yeah, for these lipids, it was not a problem.
Okay. Thank you.
For sure.
So at this point, I see no more questions in the chat. Is there somebody still waiting in line to speak up? All right, then I turn back the steering wheel to the board, and then we can perhaps conclude with the session.
Okay. Well, thank you and thanks everyone for all of you for a great set of talks. So if there are no more questions, John had to leave, unfortunately, so I will give his thanks as well for you being here for this meeting. So if there are no more questions, or comments, I will conclude the meeting.
Thank you, everyone.
Okay.
For
Thank
coming.
you.
Thank you.
Thank you very much.
End it with a gavel.